My name is Naomi Furst. I am a doctor. I'm a consultant oncologist. I specialise in the treatment of brain tumours, both primary brain tumours and secondary brain tumours. I work at UCLH, based in the radiotherapy department, and also at Queen's Square at the National Hospital. So one of the most difficult clinical problems we have is the management of glioblastomas, which are the most common primary brain tumours. These are highly aggressive, and even in the youngest, fittest patients that have the best surgery, most intensive radiotherapy followed by chemotherapy, we still only have a median survival of 14 months from diagnosis, and only 5% are still alive at 5 years. We want to improve survival, and we want to see how using better imaging techniques we can do this. What we want to do is have novel ways of imaging that help us define how much tumour is left behind after surgery. We also want novel imaging to be able to tell us how we should optimise our radiotherapy target volume. We also want to be able to get better information on how tumours are responding to treatment, both early on during treatment and at the end of treatment. We also have trouble when imaging these glioblastomas, as sometimes results can be confusing, and the standard methods of imaging that we have using MRI sometimes cannot differentiate between true tumour progression and false tumour progression. I will now hand you over to my colleague, Professor Brian Hutton, who will tell you about the more technical aspects of the imaging.
My name is Brian Hutton, and I lead the physics research group here in the Institute of Nuclear Medicine. What I want to do is to introduce you to what I think is an extremely exciting project. In nuclear medicine, we use radiopharmaceuticals to image disease and to get a distribution of the activity in the patient. And to do that, we use tomographic techniques such as single photon emission computed tomography, or SPECT, as well as positron emission tomography, or PET. Usually these techniques nowadays are combined with anatomical modalities such as CT or MRI, so that we do in the department here have PET-CT, SPECT-CT and PET-MRI. But to date in the world there are no clinical SPECT-MRI combinations which could give unique clinical information. The exciting thing in this project is that in the last 4 years, we have developed in a European project the world's first clinical SPECT MRI system. And so this project would involve you with a team of other people in really developing that system for its first clinical use. A particular feature of this type of system is that it is stationary as opposed to conventional SPECT, where the system rotates around the patient. That lends itself to being able to acquire dynamic data, and from that data being able to extract parameters which may further aid the patient diagnosis and evaluation. We anticipate that the project will be focused on developing specific software that can process data and give optimal results for clinical use. Clearly, here at UCL, we have a tremendous breadth of expertise, from expert clinicians through to expert scientists. In this particular project, we will be the first in the world to use this technology, and therefore are in a fantastic position to exploit the technology and to have high impact publications arising from that work.